So today we are going to study urea cycle. It is also known as Ornithine cycle and Krebs-Henseleit urea cycle. Ornithine cycle was the first product or first member of this reaction, due to which it is known as Ornithine cycle. And it is synthesized. Urea, as we all know, is synthesized in liver and transported to kidneys for the excretion in the urine. And it was the first metabolic pathway which was being elucidated. In this cycle, two nitrogen atoms of urea are derived from two different sources, one from ammonia and the other is from alpha amino group of aspartic amino acid. And the carbon atom is supplied by carbon dioxide. Urea is the product which is an end product of protein metabolism, or we can also say that amino acid metabolism. It accounts from 80 to 90% of nitrogen containing substances. So it is the major excretory product in urine. Urea synthesized in a 5 steps cyclic process, and in which these 5 steps 5 different enzymes are involved, among which the 2 enzymes are present in the mitochondria and the rest of them are present in cytosol. This is urea cycle. It starts from the ammonia combined with carbon dioxide in the presence of first amino acid. First enzyme. We will discuss all these enzymes in the detailed step of urea cycle, which is being then converted into carbamyl phosphate, which then combines with ornithine to form citrulline. Citrulline, in the presence of aspartate, aspartate is changed into fumarate, and Arginosuccinate is the product of further reaction. Arginosuccinate is then converted into arginine, and arginine is converted into ornithine and urea which is being secreted by the urine. So in first step, where there was conversion of carbamyl phosphate from ammonia and carbon dioxide, carbamyl phosphate synthase 1 enzyme is involved, which is a mitochondrial enzyme, and condensation of ammonia and carbon dioxide occurs in this step. 2 ATP are used. This step is rate limiting step and is irreversible in nature. The enzyme carbamyl phosphate synthase requires N-acetyl glutamate for its activity, and there is another alternate of this enzyme, that is carbamyl phosphate synthase 2, which is involved in pyrimidine synthesis, and it is present in cytosol. So it accepts amino group from the glutamine and does not require N-acetyl glutamate for its activity. The formation, this is the complete first step, in which carbamyl synthase in the presence of N-acetyl is forming carbamyl phosphate, and ATP, 2 ATP are forming 2 ADP and inorganic phosphate ions. So this is the difference between CPS1 and CPS2. You can rewind it for your cycle, or you can leave this part. The next step is formation of citrulline. It is being formed from carbamyl phosphate, and it is the second mitochondrial reaction, in which the enzyme involved is ornithine transcarbamylase, and ornithine is regenerated and used in urea cycle. Ornithine and citrulline are the basic amino acids, and citrulline is transported to the cytosol by a transporter system. Citrulline is neither present in tissue proteins nor in blood, but it is present in milk, and it is never found in protein structure because it lacks codons. So this is the final reaction of step 2. Next is formation of arginosuccinate, and it is being formed from citrulline condensed with aspartate, resulting in formation of fumarate as well as arginosuccinate. The second amino group of urea is incorporated, and ATP is cleaved into AMP and phosphate ions, and we require 2 high energy bonds in order to break the ATP into AMP, immediately broken down into inorganic phosphate. Formation of arginine is the next step from arginosuccinate in the presence of enzyme arginosuccinate liase, and which cleaves the arginosuccinate to arginine, and fumarate, which is a connecting link with TCA cycle. In this very starting, I told you people that it's Krebs-Henseleit cycle. This is the reason it is called as Krebs-Henseleit cycle, because fumarate, which is the connecting link with TCA cycle or gluconeogenesis, is formed in the fourth step of urea cycle. And the fumarate is converted to oxaloacetate via fumarase and malate dehydrogenase enzyme used to form malate from oxaloacetate and to form oxaloacetate. Oxaloacetate is forming aspartate, which is regenerated in this reaction. The last is the fifth final enzyme which is involved is arginase, which forms urea and ornithine. Ornithine is regenerated and enters again into the mitochondria for the reuse in urea cycle, while the urea is being excreted into the urine. Arginase enzyme require some metallic ions like cobalt and manganese for their activation, and ornithine and lysine compete with arginine. This is due to the process that is competitive inhibition. This enzyme arginase is mostly found in liver, like rest of the four enzymes involved in urea cycle. Arginine synthesis may occur on varying degree in many tissues, but only the liver can ultimately produce urea. And this is complete urea cycle. If we can see that first step, carbamyl phosphate synthase, then on which is forming our reaction that is carbon dioxide and ammonia combined to form carbamyl phosphate, which is then forming citrulline, aspartate and arginosuccinate, ultimately resulting in the formation of ornithine and urea. Urea is being excreted through the urine. So if we summarize the whole reaction, ammonia and carbon dioxide has undergone the reaction, aspartate was involved in the third step of the reaction and resulting in the formation of urea and fumarate. In the very first reaction, 2 ATPs were used, and in the third step of the reaction, AMP was involved, and the urea cycle consumed 2 high energy phosphate bonds. Fumarate was formed in the fourth step, which was converted into malate, and it was the connecting link between Krebs cycle. Malate was oxidized to oxaloacetate to produce 1 NADH, which is equal to 2.5 ATP or approximately 3 ATP molecules. So the net energy expenditure is 1.5 energy phosphates, and the urea cycle and TCA cycles are interlinked. Therefore, it is also known as urea bicycle. This is urea bicycle, and we end this with urea cycle only, because urea bicycle is not involved in syllabus. Thanks for listening.